Okay, good morning, everyone. How many of you had your lab practicals? Oh, you're sitting there relaxed. I can see a few more smiles today than Monday. Monday, you guys were pretty tense. Okay, for those of you getting ready, again, it's just a little epinephrine. Don't worry about it. You know, embrace the rapid stress response. Mike wanted to let me fill you guys in on a very critical information. Your final is May 7th, 8 in the morning. Bio 1A always gets the first final, and there's good and bad aspects to that. And it's 100 Haas Pavilion, which is the basketball court. One thing, it's very important here, very, very important. As per university policy, it is going to start at 8 o'clock sharp, not 8:10. Okay, we got that? Let me repeat that. Not 10 after. I know we're all programmed to do 10 after stuff, but it's right 8 o'clock on the dot. So seating will start at 7:50. Reserve seating will start at 7:50. Okay? I think you have names. You have your name tags there or something you have to go to. Okay, so that's happening there. In case you're wondering, of my 140-some points, I don't know how many points I have, somewhere around there, I've written exactly 10 points worth at this point. So as I say to you guys, start thinking about studying for the final. Same thing to me. I'm starting to think about writing the questions. And again, they'll all be one unambiguous answer to every question. And no, none of the above, all the above, because I hated those questions in college, and I still hate them now. Okay. I have an office hour today, 11 to 12, 28 VLSB. There'll be one Friday right after the lecture. I'll have a Q&A, no office hours next week, except for a Q&A review session Wednesday, 10 to 11:30 and 2050 VLSB. And you can always send me questions by email. If I feel like a question is interesting enough, or some of you have it, I'll send out an answer on B-Space. I will send out my practice questions. What is today? Wednesday? Probably by tomorrow. I figure most of you guys are worried about the lab practical anyway. You're not going to really be looking at it. That's a reasonable assumption, right? Yeah. Okay. So in the next day or two, I'll send out my practice questions. Okay. Review. Know cardiac muscles in the heart. Know what autorhythmic cells are. They contract without nerve impulses. As we talked about, they actually produce an action potential. So it's not just nerve cells produce action potentials and work by that. Know the difference between the sinoatrial node, which is the pacemaker, which is sets the rate and timing. It's also affected by adrenaline or epinephrine. So if your heart's beating a little fast right before the lab practical or the final, just blame it on that. Okay? And then you have the atrioventricular node, which is a relay point. So we talked a little bit about that. Know what those are. Lymphatic system. It defends against infection. It's obviously works with the immune system in a lot of ways, or as part of it. Recycles fluid and protein by way of capillaries. Transports fats, and has lymph nodes. You guys have many lymph nodes, and you should have some sense of that. Blood vessel structures. Know the difference between a capillary and the other structures, which are arteries, arterioles, venules, and veins. I briefly was going over the structure. The endothelial layer is the simple squamous that surrounds the capillary. So it's a simple squamous, and it has a basement membrane. All blood vessels have that. The question is, what's beyond that? So the bigger vessels have smooth muscle and connective tissue, so you can handle the pressure and you don't leak. Capillaries don't want to handle the pressure, and they want to leak. Okay? And that's how it works. Questions on those aspects? Any questions? Okay, good. And then we talked about blood composition, in particular blood cells. We'll continue with some of this discussion today. You have blood cell differentiation. You have bone marrow stem cells. What kind of stem cells are there? Are bone marrow stem cells? Multipotent, right? Some of the old textbooks have them listed as pluripotent. They are not pluripotent. They are multipotent in the current definition, because they form lymphoid stem cells or myeloid stem cells, which are two different lineages. The lymphoid stem cells give you B lymphocytes, which produce antibodies, T lymphocytes, which are part of the cellular immune response, which we will get to on Friday. The myeloid stem cells produce red blood cells, platelets, neutrophils, monocytes, eosinophils, basophils. You should have an understanding of the basics of those cells. So if I said to you, no mitochondria, which cells that I have on the board there have no mitochondria? RBCs, there we go. That's the kind of information you should be thinking about. And also some of the functionality. There are more leukocytes than what I have up there. These are just some of them and some of the more important ones, but there are others. Leukemia, proliferation of abnormal leukocytes. And you have to know the difference between chronic, and acute. Acute being an increased proliferation of immature, chronic being an increased proliferation of abnormal mature leukocytes. Acute ones, you can imagine, are more younger people and kids get the acute leukemias. There's two different lineages coming out of the bone marrow stem cells. Those define two different types of leukemias. The lymphoid or lymphoblast, myeloid or myoblast, there's other names, but it comes from those two lineages. So in essence, just by this designation, there's 4 different kinds of leukemias. You have acute or chronic, lymphoid or myeloid-derived leukemia. So the word leukemia really is a generic term. It's like saying, what did you drive to school today? For those of you who drove. And if you say a car, that's way too generic. Okay? So when you say leukemia, it's a general term for multiple kinds of problems. Questions on that? Okay. I'll leave this part up just for now because we're going to talk briefly about regulation. There's an interesting hormonal regulation, and that is when you have low, we'll get to respiratory gases here in a second, but you have low tissue and blood oxygen, because this stimulates the production of a hormone called erythropoietin. Which is made from the kidneys, near the kidneys, I should say. And what this hormone does, this hormone is actually produced in very low concentrations. And this particular hormone is one of the more, was only discovered, I think, about maybe 25, 30 years ago. It was fairly hidden in a lot of ways. This causes the proliferation, induces the proliferation and differentiation of the bone marrow stem cells into the myeloid branch, producing an increase in red blood cells. And as you know, red blood cells, as we'll talk a little more today, transport oxygen through hemoglobin. And as a result, you increase the blood oxygen. And when you increase the blood oxygen, it's a classic negative feedback system. So increase in blood oxygen reduces erythropoietin. So if you have too little oxygen, you make more erythropoietin. Actually, the other name of this is EPO. That's probably what you read about in the newspaper. I'll tell you why that is in a second. So with low oxygen, make more EPO. Down through the pathway, you make more red blood cells. You therefore increase blood oxygen, which then feeds back and knocks down EPO. If that gets too high, too low levels of blood oxygen, you produce more. So it's a classic negative feedback system. Erythropoietin is going up and down to regulate the oxygen to stay within a window of acceptability. So the idea is to keep it constant. Now this pathway is interesting for 2 reasons. One is they figured out erythropoietin came from near the kidneys because kidney patients, all of a sudden got striking anemias. And anemia means low reduced red blood cells. And they couldn't figure out why. Then they figure out erythropoietin is produced near the kidney. So there was a connection there that was unexpected. And secondly, back in the 60s, when I was in college, the big issues like in the Olympics where athletes would save their red blood cells and a couple days before their competition would shoot themselves up with their own red blood cells so they had better oxygen capacity. This particularly for events such as long distance running, things of that sort. Okay? But then they found EPO. And now what you can do, what is tried, is you simply inject a little bit of EPO into you, and then you do the same thing. So you don't have to go through the trouble of isolating your own red blood cells and putting them back in. You can just take EPO, and that will increase red blood cell production. So again, you are your hormones. It's another classic example of that. So one of the things they'll screen for is your levels of EPO, because you make very little of it. And if you have an unusual amount of it, then they know something funny is going on. Okay? And this is why it's there. And sometimes you'll see that in the newspapers. I don't know how much of that's done now, but I think 10, 15 years ago, there's quite a bit of it. They're still doing EPO now. Okay, thank you. So they're still doing it now. So the point is, this is how it works. And because it's a hormone, it dissipates fairly quickly. So I think if you time it right, you can probably increase your oxygen content. Okay? And that just happens. You discover something more about the pathway, and you can change what you can do with it. And I point that out because as you guys get into medicine, you'll be manipulating physiology through different hormonal, a lot of you through different hormonal regimens. Okay? Questions on that? All right. Now, blood clotting. I think we'll do that here. Let me see. Do I need this? No. Blood clotting. Just very briefly before we get into respiration. It's always good to talk a little bit about blood clotting. And what happens is, here's a vessel, and you get an injury, and you break open the vessel itself, and you start bleeding a little bit. And if you have little kids or lots of kids, you have to carry Band-Aids at all times because they're always falling and injuring themselves a little bit. And what happens is you form a plug, platelet plug is the first thing that happens. To kind of stop it, what it is it recognizes the connective tissue that gets opened up and binds to it. And as a result of this, you've got different kinds of, you got platelets around, which are fragments or pinched off fragments of the myeloid stem cells. You also got cell debris, and you have various ions, calcium, for example, which we won't get into. So so when you open up, when you start this platelet plug in the bloodstream, you cause some extra material that gets in there. Now I'll show you what happens to that. What happens to that is you activate a protease called thrombin, or prothrombin, which is a precursor that's larger, and it makes thrombin, which is an active, let me just write this a little clearer here, which is an active protease. And what activates this is the combination of cell debris and ions that get produced. In addition, there's a huge cascade of protease. There's a huge protease cascade. Don't want to get into the details, which ends up with a protease that cleaves prothrombin, because this precursor is larger, splits off a little bit of the end of it, and you get thrombin, which is active. And what's interesting about this protease cascade, there's a factor in the middle of it called factor 8. And that is the factor that is genetically missing or defective in people, in hemophiliacs, certain classes of hemophiliacs. In particular, all the kings and queens and princesses in Europe, they all had hemophilia. A lot of them had hemophilia. In that, because they were cross-fertilizing with each other, so to speak, a lot of these recessive genes stayed in the gene pool, and what they're missing was factor 8. So this protease cascade kind of was very, very dampened, and you didn't really produce much thrombin from the prothrombin, and they became a hemophiliac. And you'll see why that is in a sec. Okay? So that's why that's important. Now, thrombin, which we'll do over here, has one substrate, one particular substrate. And the substrate is fibrinogen. And through the actions of thrombin, you make fibrin. And what fibrin does, it goes up to this opening in the connective tissue and forms a clot, the fibrin clot. And the fibrin clot is what you see ends up being your scab when it dries out and everything like that. So the clotting process is you injure yourself, you have cell debris, which stimulates the system, tells it there's something wrong. The protease cascade comes in, converts prothrombin to thrombin. That protease has one particular substrate, fibrinogen, goes to fibrin, which forms the clot and, in theory, stops the bleeding. Okay? Now, you ask yourself the question, we talked about basophils. Basophils prevent clotting, but it only prevents it when you don't have any injury. Because if you clot without an injury, you get an internal clot, and that could lead to a stroke. But when you're injured, you want a clot. So this system kind of overrides any effects of the basophils of trying to stop clotting, because there's not that much heparin that's produced. And this completely overrides it and allows you to clot on the surface. Okay? Questions on that? Good. Okay, what ties into the circulatory system is respiration. As we continue our systems flyby here. And really what respiration is gas exchange. Oxygen in, CO2, carbon dioxide out. And what happens is, in the deep recesses of the lung, you have these air sacs called alveoli. Actually, here's the air sacs over here. I'll make it a bigger air sac. Alveoli, which is an air sac. And these are directly connected to the lung capillaries. And there, what you have is, what you want to do is get rid of CO2, and you want to bring in oxygen. Simple kind of concept. But there's actually some gory details in there that are worth thinking about. Now, in gas exchange, the way to think about gas exchange is these gases move from high to low pressure. And this is the overriding force, what dictates whether oxygen goes in or out or CO2 goes in or out in a given situation. So let's think about some numbers. You don't have to memorize these numbers, but I want to give you a sense here. What is the atmospheric pressure at sea level in terms of millimeters of mercury? 760, right. So your air pressure is 760 millimeters of mercury at sea level, okay? Which is basically here in this room. Now, oxygen is about 21% of the atmosphere, and therefore the partial pressure of oxygen, or PO2, equals about 160 millimeters. Well, forget to worry about the millimeters of mercury, just so that's what it's all based in. In contrast, now this is in the air. The partial pressure of CO2 in the air is much smaller, is about 0.23. So you can see that we have a lot more oxygen than we have CO2. But in the alveoli, remember, you're getting rid of CO2, so you dilute it out, or you dilute it out, the concentration. You increase CO2 concentration, decrease O2. In the alveoli, or in the lung capillaries, say the alveoli, the partial pressure of oxygen is about 104. Again, you do not need to memorize that number. You just need to know where it's high versus low. And partial pressure of CO2 is about 40 in the alveoli. So it's coming into the lung capillaries here. Here's the lung capillaries. The partial pressure of oxygen to CO2 is 104 over 40. Well, these are the numbers we're going to be playing with here. So the top number is going to be partial pressure of oxygen. Bottom number is partial pressure of CO2. Okay, everybody with me there? Okay? So what comes into the lung capillaries from the heart, remember you have the heart hanging out here, and you got the pulmonary circuit. Okay, blood going into the lungs, is that oxygen rich or oxygen poor? Based on what we talked about Monday briefly. Oxygen poor, right. Because the O2 to CO2 ratio is approximately 40 to, say, 45. So partial pressure of oxygen is 40. Partial pressure of CO2 is 45. What comes into the lung capillaries, it sees in the alveoli 104 to 40, oxygen over CO2. So when this oxygen-poor blood is coming in to the lung capillaries, and it sees 140 to 40, what does the oxygen do? Remember, you move from high to low pressure. So the alveoli, it's 104. It sees blood at 40. So how does the oxygen move in terms of high to low? From the alveoli into the blood. Does everybody see that? Because you're going from 104 to 40. Where does the CO2 move? From the blood out, because the blood coming in is 45 and the alveoli is 40. So in the lung capillaries, O2 comes in to the blood, CO2 out. Simply because 40 is much less than 104 and 45 is greater than 40. Everybody see that? That's all that's going on there. So now you have this oxygen-rich blood. And on the other side of the heart, let me do the other side down here. On the other side of the heart, now we're coming into the tissues. This goes back into the heart. And now we have the systemic circuit. Actually, I should write the word heart because H has been hormone most of these lectures. So there's no confusion here. Now we go to the systemic circuit. And this, coming from the heart, it comes from the lung into the heart, back out to the system. So it stays at 104/40. So it's oxygen-rich. So it goes into the tissue capillaries. And in the tissues, even if just sitting there and listening to a lecture, your partial pressure of oxygen is 40 and CO2 is 45. So it comes in to the tissue capillaries in the systemic circuit. It sees oxygen at 104. It's coming in at 104, right? Because it's coming from the lung. It sees the tissue. The tissue, partial pressure of oxygen is about 40. So how does oxygen move if you go from high to low? Oxygen should go in, right, to the tissues. And what comes from the tissues back into the circulation? CO2, because CO2, it sees 40, but it is 45. Exactly right. So what comes out of this is now oxygen-poor blood of approximately 40 to 45. And that's how it works. That's how the system works. In the pulmonary circuit, you take oxygen-rich poor blood, turn it into oxygen-rich because of partial pressure differences. Oxygen goes in, CO2 goes out. In the tissue capillaries, of which there are many, the oxygen-rich blood comes in because of differences in partial pressure. O2 goes into the tissues, CO2 comes out. So it all works by going from high to low pressure. That's the way to think about it. Question?
What happens to the partial pressure of oxygen in the lungs after blood leaves the lungs?
Now, the blood never leaves the lungs. It's a continuous flow system.
Yeah.
Okay, it's like turning on a spigot in a garden hose and you're running your water. Water never is leaving that hose. So it's a continuous flow. So there's always blood going through the capillaries. You exhale out the CO2 and you inhale the oxygen. Because you're exhaling out CO2, even though the air partial pressure is 160 or something like that, by the time it hits the alveoli, it's only about 104. So you're diluting it out.
So because we have like continuous blood flow and because we breathe in and out all the time, in the lung capillaries, the partial pressure of oxygen remains as 104.
Correct. The blood leaving the lung capillaries is 104. The blood entering the lung capillaries, partial pressure of oxygen is 40.
Yeah.
So you're coming in at 40, leaving at 104.
How come the capillaries leaving the lung is so high?
Why is the oxygen so high? Because you're breathing it in from the air.
Oh, I
See, what are alveoli? They're air sacs. How do you fill those up? From the air. Right.
If the gases are dissolved in the blood, then how could the partial pressure?
Pardon? Say again?
If the gases are actually dissolved.
Yeah, we'll talk about how it's transported. Gases are not totally dissolved. You'll see that in a second.
Okay.
Okay? Now, in terms of carrying this, what's responsible for carrying the oxygen is the red blood cells. And in particular, in the red blood cells, RBCs, you have a protein called hemoglobin. And it carries the oxygen. It directly carries the oxygen. Okay? And so you have 4 subunits. So there's this interesting thing to think about here. Here's the 4 subunits. A little bit of oxygen comes in, binds some of it. The binding of some oxygen increases the affinity for the additional oxygen. So you bind one, and all of a sudden you have a much higher affinity. And you quickly fill up the rest of it. You saturate the hemoglobin. So you become quickly saturated. And this concept of increasing the affinity is given a term, you guys may have seen this before, called positive cooperativity. Let me just put it here. Simply means, in this case, binding of one molecule increases the affinity for the other molecules. You want this to happen because when you're in the lung capillaries, you're being inundated with oxygen. You want to quickly bind as much of it as you can. You want to totally saturate the hemoglobin because that would allow for efficiency in the system. Does everybody see this? But you also have to get rid of the oxygen. And you get rid of it and it comes off based on the partial pressure of oxygen. So the lower the partial pressure, more oxygen released. So if that hemoglobin, for example, to your question about the blood flow, if its blood stopped flowing and it stayed in the lung capillary, you wouldn't release much oxygen because the partial pressure is so high. But when you're in the tissues, because the partial pressure is much lower, you release the oxygen. So this release from the hemoglobin is based on it. There's slightly more to it than that, and you'll see that in a second. But that's the fundamental concept. Yes?
So if we stop breathing and actually stop the blood flow, would the partial pressure in the capillary blood and the alveoli become the same?
The question is, if you stop breathing, would you start to equilibrate? The answer is probably yes. Okay? But that's not a good situation. Okay? Maybe if you're a Laker fan getting ready for the playoffs, that might be good, but not for the normal situation. It's easier than breathing, maybe.
How do the blood carries from the lung to the toes?
Well, we're not going to get into that, but you have the movement of the smooth muscles and stuff. We'll keep the blood going. We're not talking about that. Okay. Now, there are some ways of thinking about this in the sense of people have quantified this quite a bit. And one quantification is, if you look at the partial pressure of oxygen, there's a couple key numbers here, 100, 40, and 10. And this is saturation of hemoglobin. Where is hemoglobin saturated, at 10 or 100 partial pressure? Where's it saturated, 10 or 100? 100, right. That's a good answer. Okay? So this is the lung capillaries. You should recognize this partial pressure as approximately what's in the lung capillaries. When you guys are sitting there under normal metabolism, and let's just say you're not completely stressed out, but you're sitting there listening to the lecture, your partial pressure is about 40. So this is the oxygen release under normal metabolism. obviously this will differ slightly from person to person. But maybe some of you decided to run 10 miles or 5 miles before you came into the lecture. I don't know why you'd ever want to do that, but I certainly couldn't, even if I wanted to. Your partial pressure of 10 is active metabolism. And so the amount of additional oxygen that you can release, additional O2 release when you're active. So if you're just sitting there casually walking to lecture, you're probably using up this much oxygen. Then all of a sudden, you decide to just run out and run a mile to your next class. Maybe I accidentally end the class 20 minutes late, which I would never do. And you have to run to the next class. You use up this additional oxygen. So you have this oxygen reserve. Yes?
So this partial pressure is why people have difficulty when they go at high altitudes in the air?
Yes. I'm not going to talk about this, but this whole concept of partial pressure, everybody responds a little differently. If you go up to high altitude, what's the partial pressure up in the Himalayas? Is it 160? Is it greater or is it less?
Less.
It's a lot less. So this whole system is shifted down. And you have ways to correct for that. But it takes days. To change, you produce a molecule that increases the affinity of hemoglobin for oxygen. So you can, the little bit of oxygen that's there, you can take it. That's why it takes a while to adjust to high altitude. That's when I go up to high altitude, I get altitude sickness for a couple days, but then I'm okay maybe 2 days later. You can adjust and increase the affinity. That's exactly correct.
So what happens when like a team which has trained at high altitude is like playing against a team which has,
Yes, that's right. You can, like, for example, sporting events, we have to do a lot of exercise or exert a lot of exercise. Or say people taking the SATs can only get it up in Tahoe, so they have to go up there to take their SATs, for example, for use to sea level. Same kind of use of oxygen. Yes, there is issues along those lines. For the Mexico Olympics in the 60s, Mexico City Olympics, people would take entire teams and train for a month at the high altitude prior to the Olympics, so they get used to it for exactly these kinds of reasons. Okay? You can also do is there's some hormones you can use also, which are not legal, to change this. You don't want to do that. The other thing too is like the Himalayas, people are mountain climbers, right? Mm-hmm. Some people need that oxygen sooner than others. People that are from that area almost don't need it hardly at all. So there's those kind of issues. But it's all related to this capturing of oxygen by hemoglobin and releasing it under activity. Now, some of you say you want to go run a marathon. I mean, I have a couple, or a half marathon or something like that. And you have another system that relates to this. Here's your partial pressure of oxygen. Here's 10, 40, and 100. Now we're talking about extreme exercise. So here's saturation of hemoglobin. So here's, normally you're about pH 7.4. What will happen is, for extreme exercise, this entire curve gets shifted a little bit to the right. And this is when your blood acidifies to 7.2. And some of you can feel that if you run a lot. For example, don't you feel all of a sudden your muscles you're getting pain and stuff? That's from the acidic conditions and everything. This is when it starts happening. What this allows you to do is, at this extreme exercise, if you look at these, where these numbers come out, you have this little additional oxygen release that's available. And it's kind of related to, if you're running like a marathon. I've never run one, nor will I ever. But you get that sort of like second wind that takes place. If you get enough, go acidic enough, this extra oxygen helps out. People think that's an evolutionary, from evolution, because when humans were going after game, they'd go for an animal for their food, but they'd only wound it, so they have to chase it for 3 or 4 days. And this added oxygen allows them to have that extra energy over long distances to do that. So that's a real phenomenon. It's called, and the name of this shift from low to high pH is called the Bohr effect. Okay? This is all related to the ability to saturate hemoglobin and the ability to release it at certain partial pressures. Now the question is, to what some questions earlier in this discussion, how do you transport oxygen? If I said to you transporting oxygen, how do you transport oxygen? As a free gas in your bloodstream? No. As another molecule? No. How do you transport oxygen? You transport it bound to hemoglobin. That's how you're transporting the blood. This is in striking contrast to CO2. Okay? And for CO2, we'll go back over to here. The CO2 side of this is really quite different. So let's start with the lung. Let's start with the tissue capillaries, rather. Is CO2 high or low in the tissues after a little bit of exercise? CO2 high or low? High-ish, right? So what happens is the CO2 from the tissue capillaries goes into the tissue capillary, and it goes into the red blood cell. CO2 is then combined with an enzyme called carbonic anhydrase. And water to give you H2CO3, carbonic acid. You remember a couple days ago I said any enzyme that ends in ASC usually means what? Breaking something. In fact, this does break stuff, but this is the reverse reaction. Some enzymes are reversible, and this is the reverse reaction from how it was discovered. We'll see how this works. Okay? So let me just put this up here. This is all still in the tissue capillary. The carbonic acid then, a proton splits off. And the proton goes and binds the hemoglobin. This binding of the proton changes the structure and allows the hemoglobin to massively release the O2, the oxygen. So it's carrying oxygen to the tissue capillaries. It's CCO2. CO2 gets converted into carbonic acid by the enzyme carbonic anhydrase. The proton comes off, forming bicarbonate and a proton, and this binds the hemoglobin, releasing the oxygen. In addition, a little tiny bit, and just a little tiny bit of the CO2 binds to the hemoglobin, but not much. So it's a regulated system. So what happens is the bicarbonate then goes out into the bloodstream. Therefore, one transports CO2 as bicarbonate, not as a gas. A little tiny bit of it goes by way of hemoglobin, but mostly it gets transported as bicarbonate as an ion out in the bloodstream. Okay? Then now we're in the lung capillaries. And here's the same red blood cell, or it could be a different one, I guess. You've got many. Bicarbonate comes in. Because the partial pressure of oxygen is so high, you start flooding this thing with oxygen, and it causes the hemoglobin to release the proton, which comes back and forms H2CO3, which is what? What's H2CO3? Carbonic acid, right? Carbonic acid. Then, this H2CO3 is the substrate for carbonic anhydrase. Same enzyme. Which is the way this enzyme was discovered, because it cleaves the bond in carbonic acid, forming CO2 plus water. And then the CO2 goes out the lung capillaries to the alveoli, and you exhale it. So the same enzyme is responsible in the tissue capillaries for forming bicarbonate. It's the same enzyme that breaks it back down into water and CO2. So basically, you bring in CO2 from the tissues, gets converted to carbonic acid, the proton comes off, hits hemoglobin, causes the release of the rest of it. That's why the Bohr effect works so well. This is the acid from part of the Bohr effect, that you release that final little bit of oxygen. The bicarbonate gets transported through the blood. Little tiny bit of it gets transported in hemoglobin. This then sees the proton because of the oxygen uptake. In the lung capillaries, you get carbonic acid. Carbonic acid then, make this a little bigger here so you know it's the same molecule, gets hit by carbonic anhydrase, and the CO2 goes out. And the water, of course, goes back into the circulation. Questions on that? Any questions on that? Yes. One.
How does carbonic anhydrase determine whether it catalyzes the formation or the breakdown?
Yeah, how does it know? How does the very good question. How does the enzyme know what direction to go?
From the reaction?
Who's ever the highest concentration. So if CO2 and water are around, it goes one direction. If there's a lot of carbonic acid around, it goes the other direction. So it depends on the substrate concentrations. Again, every self-respecting enzyme must have a substrate. In this case, interestingly enough, the substrates and the products are both products and substrates. So it depends on the relative concentration. Okay? All right. Shifting gears, excretory system. Kidney function. And we'll start in on that. Again, here's a system where the idea of capillaries forming an equilibrium with interstitial fluid and with cells is critical in the understanding. So I'm going to give you the generic system first before we go into the details. Basically, you have blood flow, and you go into some capillaries. Make this even bigger here. The kidney capillaries. You'll see what those are in a second. These capillaries are semi-permeable. So it is an example of capillaries that it's not a complete free-for-all. Only some molecules are transported through, or allowed to equilibrate through. Now, in chemistry, if you're filtering something, so it's like a filter, and you have those. I know I'm drawing upon nightmares of organic chemistry, but after you filter something, what's that stuff called after you filter it? I mean, it's liquid. Filtrate, right? That's exactly what happens in the kidney. These semi-permeable capillaries forms a filtrate. The reason is in the kidneys, in the excretory system, you want to get rid of waste and keep nutrients. And eventually you want to make urine, which is concentrated in terms of molecules. And in this system, you also have a recycling part of it. You have reabsorption from the filtrate back into the blood, and you also have direct secretion in some places, actually through capillaries, through interstitial fluid, and back into the cells. This is what's going on at all times. So you go through the interstitial fluid, and you go back in the urine, so you get rid of waste, some waste, and the reabsorption, you produce and recycle nutrients. An example of a waste would be like ammonia. You want to get rid of that. An example of a nutrient might be, say, sodium chloride or some sugar that gets through. So this is the generic system. The blood comes in. The capillaries are semi-permeable, meaning they're just not completely leaky. They're selectively leaky. The filtrate comes through, and you've got to clean it up a little bit. You've got to clean up your mistakes. And you reabsorb nutrients, and you get rid of waste. Now, there's more to it in this, as we're going to get into, but this is the generic part of this system. And this is all based on forming an equilibrium between the capillary, interstitial fluid, and cells. The big issue is urine is about 4 times concentrated than the blood in terms of molecules. And it's how it does this is the major issue in kidney function. So first, and the way to think about this is simply a bunch of plumbing. The functional unit is a nephron and a collecting duct. And we'll just do the anatomy here. We have time for that. Going into it is something called the afferent arteriole. Let me just. And the selectively permeable capillaries are called the glomerulus capillaries. Let's make this bigger here. In a structure of the nephron called the Bowman's capsule. So these are glomerulus capillaries. And this whole thing is called Bowman's capsule. You'll get a bigger view of this in a second. And this continues in with the rest of the nephron. And so this is a higher magnification of what I'm going to draw right now. So here's the afferent arteriole. Here's Bowman's capsule. And he has a structure that sort of looks like this. Okay? Right here is something called, there's a loop involved. This is called the proximal tubule, this structure here, right after Bowman's capsule. And this undergoes secretion and absorption, reabsorption. So what goes on there is this process, reabsorption and secretion. And I'll just have it on the diagram like that for now. You have this big loop, and a researcher by the name of Henle discovered it, one of the first microscopes, and this is called the loop of Henle. And what you have here is a descending limb, so the flow is from here through it. So this is the flow of the fluid. This is the descending limb. This is the ascending limb. Then you hit the distal tubule, which also has absorption and reabsorption and secretion. And then you have the collecting duct, which is the other part of this. And then you make your urine. You concentrate. Okay? So the plumbing is afferent arteriole, Bowman's capsule, selective permeability, infiltration, proximal tubule, reabsorption and secretion, bad in, get rid of the waste, keep the nutrients, go through the descending limb, up the ascending limb, go to the distal tubule, and down through the collecting duct. So the question is, why do you do this? You do this to concentrate the urine. And the whole reason why this thing gets concentrated, the last point here, because they don't want it, that each of these structures has a different function. And what it changes is the osmolarity. Have you guys heard the term osmolarity? No. What about molarity? Okay, osmolarity is the number of molecules. So what is the molarity of sodium chloride, for example, approximately in your system? What's physiological concentration of sodium chloride? It's about 150 millimolar. Have you heard that term? No? Doesn't matter. Now you have, it's 150 millimolar. But the osmolarity is double that because you have sodium chloride, but you have a sodium ion and a chloride ion, right? Sodium chloride is made up of 2 molecules. So the osmolarity is 300 millimolar. So physiological molarity is 150 millimolar. Physiological in the bloodstream of osmolarity is 300 milliosmolar. And what we'll talk about is, out here towards the blood, you go from 300 milliosmolar down to urine, which is 1,200 milliosmolar. So you concentrate the molecules in the blood fourfold for the urine. Okay? So we'll go over this concept again Friday. All right, for those of you taking the lab practical, good luck. If you have questions, we have office hours 11 to 12, also by email. Okay? We'll go over this again, the osmolarity issue.